Hello, this is Dr. Amardev Rajesh Vatapatti, and I am a bioinformatician. I did my master's in bioinformatics from SWIMS University and got my PhD in biotechnology from Acharya Nagarjuna University, Guntur. Today, I would like to present my first research article, which is in Scopus Journal, which I published in the year 2007. The name of the journal is African Journal of Biotechnology, and the full length of the research article is available in the volume 6 of African Journal of Biotechnology. The details of the full length papers I'll keep it in the description for your views. Okay. The background of this research article is from my MSc dissertation. MSc Bioinformatics dissertations. The title of the dissertation is Sequence Analysis and Protein Modeling of Apurinic and Apyrimidic Endonucleases. This I have submitted in partial fulfillment for the award of the degree of Master of Bioinformatics, the Department of Bioinformatics, SWIMS University, in the year 2005. Later, this is published as a full-length research article in African Journal of Biotechnology, which is a Scopus indexed paper. And the title of the research paper is Protein Modeling of Apical Membrane Antigens, which is otherwise called as AMA-1 of plasmodium cynomolgi. The first author is Dr. D.M. Mamata, Department of Sericulture and Biosciences, Sri Padmavati Women's University, Tirupati. The second author is K. Nagalakshmamma, belongs to same department of Sericulture and Biosciences, SPMEV, Tirupati. And third author is myself, Department of Bioinformatics, Dravidian University, Kuppam. And the fourth author is V.S. Shirin, and she also belongs to SPMEB University. And this paper got accepted in the year 26th October 2027. The abstract of the paper is as follows. Apical membrane antigens and asexual blood stage antigen of plasmodium cynomolgi is an important candidate for testing as a component of malarial vaccine. The degree of conservation of AMA-1 sequence implies a conserved function for this molecular across different species of plasmodium. Since the AMA-1 of plasmodium cynomolgi is yet to be structured, the authors have generated a homology modeling of AMA-1 by using Swiss PDB Weaver, an offline tool. The proteins conserved has been verified by performing multiple alignment using bioedit and conserved domain databases. The molecule was further checked for its correctness by predicting 2D and 3D structures, which validates the structure. Okay. Now coming to the introduction part of the paper. Most of the activities in living organisms are regulated by proteins. All proteins start on a ribosome as a linear sequence of amino acids. This linear sequence must fold during and after the synthesis so that the proteins take up its native conformation. The native conformation of the protein is a stable three-dimensional structure which strongly determines a protein biological function. The knowledge of detailed structural organization is crucial in understanding the role of a protein in the cell and related molecular mechanism. Insight of a three-dimensional structure of a protein are of great assistance when planning experimental aims at the understanding of protein function and during the drug design process. The experimental elucidation of the 3D structure of the protein is, however, often hampered by difficulties to obtain sufficient proteins. Diffracting crystals and many other technical aspects. The number of solved 3D structure is small when compared to the rate of the sequence of genes, and no structural information is available for the vast majority of the protein sequence. At this juncture, prediction methods have gained much interest. Genomic Sequence Project continuously detects new protein sequences. This provides new information for the application of computational methods, representing a good alternative to relatively slow experimental process for predicting protein structures. This study focused on building molecular models of AMA-1 protein, a plasmodium cynomolgi, using molecular modeling techniques, Swiss model server, to build a theoretical model of AMA-1 protein by homology modeling, and to understand the fundamental characteristics of AMA-1 protein by comparative analysis with the template. The model was generated using Swiss model v3.7 and automated protein model server available at xpc.org. Coming to the materials and method, homology comparative model is carried out. If the native sequence similarity with the similar crystalline or NMR structure is greater than 35. The process of building a comparative model is conceptually straightforward. First, an alignment is performed between the sequence for which the structure has been determined by experimental methods with the sequence to be modeled. The comparative modeling of AMA-1 was built using a web-based homology modeling server, Swiss model. It predicts the model in 5 steps. Firstly, it searches for suitable template for submitting the protein to be modeled in the 3D databases using blast-proof programming. In the second step, it selects the suitable template with sequence identity above 25%. Furthermore, this step also detects domains which can be modeled based on unrelated templates. Third jobs are created for Swiss model server, protein server in the fourth step. Fifth step, minimize the energy of all the predicted models with the Gromos 96 module of Swiss model server. Finally, the best 3D model having minimum energy is sent to be submitted. The sequence identity of known structure was checked with AMA-1 protein using SIM program that selects all templates with sequence identity above 25%. Furthermore, this step also detects the AMA-1 domains which were further cross-checked with the CDD, conserved domain databases. The energy minimization of the generated model was done with Gromos 96, followed by secondary and tertiary structure prediction. The multiple alignment of AMA-1 protein from plasmodium cynomolgi were generated using bioedit and conserved domain databases. The secondary structure of the model was predicted by using NPS at GOR4, predict protein server and Swiss model server. The model was analyzed for its correctness using project results and discussion. The multiple alignment of protein sequence of AMA-1 of plasmodium cynomolgi highlights the degree of the sequence similarity and high degree of sequence conservation with templates selected, which implies the conserved function for this molecule across the different species of plasmodium. Pairwise comparison between all the known amino acid sequences of AMA-1 homologous indicates greater than 50% identity with the 16 crystalline residues conserved in all sequences. All these cysteines were found in the ectodome of the molecule, which are stabilized by 8 intramolecular disulfide bonds. The ability of this molecule to induce a protective immune response has been shown to be dependent upon a conformation stabilized by disulfide bonds. The secondary structure prediction by the protein server matches almost exactly with the secondary structure of AMA-1 homology model. The overall summary: protein were classified as 24 beta sheets, 7 helix, and 27 turns. The results from Swiss model and predicted protein almost coincided exactly and also shows a high degree of conservativity to the template used. The energy minimizations were done with Bromos 96, implementation of Swiss PDB server. Further, territory structure done on the project. Z-score show 84.2% residues in the most favorable region, 13.4% in the additional allowed region, and 1.2% in generously allowed region, and 1.2% in the disallowed regions. So conclusion, using protein modeling tools, building a homology model for this protein has been accomplished and visualized. In the absence of 3D structure of AMA-1, protein of plasmodium cynomolgi, the generated protein model of AMA-1 may be used to understand the antigenic sites and regulatory function by visualizing its domains and structural conformations. The degree of conservation of AMA-1 sequence and structure implies a conserved function for this molecule across different species of plasmodium. Apical membrane antigens, which is an asexual blood stage antigen of plasmodium, is an important candidate for testing as a component of malarial vaccine. However, before using the model for further work, it should be checked for its geometrical, stereometrical, stereochemical, and conformational accuracy before taking up for rational drug design to avoid later complications. And the details of the paper and the figures and the results will be kept in the links will be kept in the description for reading. Thank you.